present on a novel small molecule drug-based approach for treating beta-globinopathies.
Right. Good afternoon, everyone. I'm Ziyang, research assistant from Brigham and Women's Hospital, Boston, under supervision of Dr. Jun and Dr. Lee from the Chai Lab. Here I'm very excited to share some new data on the project I'm working with for the past 2 years on a novel small molecule drug-based approach for treating beta-globinopathies. So this talk will be divided into briefly up some background, and then how we identified SH6, and then we'll talk about the future direction. So thanks to the previous presenter, I don't need to go into too much details on beta-globinopathies. It's a devastating disease with significant global unmet needs. Sickle cell disease and beta thalassemia patients with HPFH, hereditary persistent fetal hemoglobin, demonstrate decreased symptoms and disease-related complications. So the question is, how do we reactivate HbF? So going just a little bit into the genetics, during before and after birth, there's a switch from gamma to beta globin. And then there's transcription regulators, mainly CBTP7A, BCL11A, recently WIS, that regulates this globin change. So the strategy then is, by downregulating these transcription factors, we can reactivate HbF. So in the current landscape of the therapeutic modalities, we have hydroxyurea, the only FDA-approved gamma globin inducer, but only works on a subset of patients. We have decitabine, which also is a hypomethylating agent, but it's a global demethylator, so it has some safety concerns as well. We have exciting investigational gene therapies, which are in the clinical trials listed right here. We have FDA-approved gene therapy, such as Vertex and Casgevy, by editing BCL11A, but it's still in this early stage and it's expensive and relatively have scalability issues. And we also have investigational TPD, targeted protein degradation, such as the WIS degrader, BCL11A, PROTAC, and antibody. And our work trying to, adding to this field, we're hoping our new drug, SH6, can help fill in the gap. So how do we identify SH6? So the Chai Lab has spent nearly 2 decades on actually another protein called SAL4, which is an oncoprotein that activates in the fetal stage, but then deactivates when it becomes adult. So from 676 compounds, we use AlphaFold, crystal structure, and SAL4 zinc finger cluster 4 docking. We eventually get a lead of SH6. Turns out to be a non-IMiD molecular glue degrader of SAL4. And we did a mass spec on this drug for SNU398 cells, which is a liver cancer cells. We see a degradation of ZBTB7A. And also, we did a docking of SH6 docking against the predicted binding sites of ZBTB7A with the help of a lab from Singapore. So based on these facts, we hypothesize, does SH6 induce HbF by degrading ZBTB7A? So using HUDAP2 cell line, it's a very good cell line because it has a low HbF expression. We use a sort of a shortened differentiation scheme, so we can test different doses at a sort of very high throughput manner. And then we did flow cytometry looking at the HbF expression. So the x-axis is the fluorescence intensity of the HbF, and the y-axis is the normalized counts. So looking at SH6 here, it outperforms known gamma globin inducers, including pomalidomide, decitabine, and hydroxyurea. To confirm what we see in flow cytometry, we use HPLC as well, courtesy of Dan Bauer Lab. HPLC is a clinical grade assay that detects functional HbF tetramer. So looking at the peaks right here and here, we see that compared to DMSO, we have a induction of HbF from 2.7% to 18.9%. So we see good data from HUDAP2. The next logical step is then move on to CD34 positive primary cells. So we use a typical differentiation scheme. And we realized two more things. So we see minimal toxicity when we lower compared to DMSO and S66 at 0.05 micromolar. So here we see a sort of a cell count graph, and then we don't see really cell number changes with drug treated or not. And then we use similar to HeatUp2, we use flow cytometry to test if we can see induction of HbF, and we do. And then we also see this at nanomolar scale. Here we give a representative data of 0.05 micromolar or 15 nanomolar. And again, we use HPLC to confirm our findings, courtesy of Dan Bauer lab. We see from 10% to a 20% increase in CD34 positive primary cells. So one critique we usually get is, does the drug increase HbF by actually inducing HbF or disrupting the sort of the differentiation? So we look at these differentiation markers as well, CD71, CD36, and CD235A. And then looking at all three of those markers, we don't see a lot of changes between DMSO and the treated. So we are pretty confident that the drug, the HbF inductions actually come from the drug itself instead of disruption of the erythroid differentiation. Just dig into a little bit more into the mechanisms. Since we previously hypothesized that the SH degrades ZBTV7A, induce HbF in a cereblon-dependent manner. So in order to test that, we borrowed a lot of knockout cell lines, courtesy of Dr. Stuart Orking's lab. So we borrowed cereblon knockouts, HeatUp2, and then we use tests in qPCR and flow cytometry. So here in qPCR, we see gamma globin induction in wild type, but no longer in cereblon knockout. And in flow cytometry, we see, again, normal, like expected induction in wild type, but no more induction in the cereblon knockout. So after establishing that cereblon knockout no longer increases HbF, now we turn into if ZBTB7A is actually degraded. So as expected, Western blot results show that in wild type, ZBTB7A is degraded through so ZB7A is degraded in wild type, but not degraded in cereblon knockout. And then also, curiously, we also see degradation of BCL11A, which is also an important transcription regulated HbF induction. But the exact mechanism is unclear. We are working hard to figure out its mechanism. So in terms of preliminary toxicity study in NSG mouse model, we see after treating the drug with SH6, or after treating SH6, we see no significant weight loss and no significant changes in CBC and organ weight between groups. So in summary, we see that SH6 induced HbF expression in HeatUp2 cells and primary CD34 positive erythroid cells. SH6 induces HbF by degradation of ZBT7A and likely BCL11A dependent on the cereblon pathway. So combining those two facts, to evidence, we see that SU6 has the potential as therapeutic agent for treating sickle cell disease and beta thalassemia. So in terms of next steps, we want to do more in vitro CD34 positive primary cells, not just from the healthy, but also from sickle cell disease and beta thalassemia patients. We will also do in vivo xenotransplant with healthy donor sickle cell beta CD34 cells and drug treatment. We would dive a little deeper into the mechanism to see what the background is, because we see it degrades BCL11A, ZB7A, and potentially other protein as well. And then we want to optimize our SU6 by either increase its potency or reduce its off-target effects with collaboration from medicinal chemists. And hopefully we get more funds raising towards the IND. So thanks to my advisors, so Dr. Jun Liu and Dr. Li Chai for the constant support. And I'd like to thank Hannah and Na to do a lot of the wet lab work for this project. Thanks Cooley's Anemia Foundations for their generous support of the grants. And then this project would not be possible without the help of these PIs. So Dr. Dan Bauer, Stuart Orkins, Sean Stoll, John Manis, and Kim Van der Schild. So yes, so thank you everyone, and then happy to take questions.